Hello, and welcome to our discussion on the cardiovascular system. In this segment, we are continuing on with blood. So, this is our second part to this chapter, this topic. And last segment, we sort of familiarized ourselves with blood, saw how it was a sort of cocktail of life, so to speak. In other words, blood is a colloid that contains many special substances that help cells in the human body do their essential functions. In this segment, we look how that blood then sometimes clots and can do a repair in tissue. And then also we're going to look at groups of blood and how transfusions might come into play. As always, these will cover the highlights of our assigned course readings and as important concepts, noting that stopping the bleeding is a very important homeostatic mechanism. This first slide is sort of a general overview of what we're going to be talking about over the next few minutes. Notice on the left we have a damage, a homeostatic imbalance. Notice there's a incision placed in that integumentary system. So inflammation is going to set the stage. It's going to begin. Notice that these white blood cells are migrating towards that area of damage. Well, with white blood cells migrating in, doing their work, we're able to regenerate some epithelia. Recall that integument has a substantial amount of collagen fibers. We're going to build these collagen fibers to help repair the wound. Lastly, we're going to get a quote-unquote scar, so to speak. We're going to regenerate some epithelium. The epithelia regenerates and underlying area scar tissue results. So we're going to go more in depth into this process. And we start with the topic of hemostasis. And we might call this a process whereby blood clots, and it's also going to inhibit the flow of blood through an injured vessel. And here this diagram shows the general steps. Generally speaking, first we're going to get a vascular spasm. That means we're going to get some smooth muscle contracting inside the cell walls, the blood vessel walls, causing vasoconstriction. The next thing we're going to see in hemostasis is the development of a platelet plug. And then thirdly, this idea of blood clotting, that's also called coagulation. This is where we're going to get fibers such as fibrin to form a sort of mesh that traps the red blood cells and platelets, forming a clot, thus preventing the loss of blood. So let's go more in depth into that vascular spasm now. So this vascular spasm is a process by which a blood vessel constricts suddenly. So this sudden constriction is going to restrict the amount of blood leaking from that damaged vessel. And this can reduce blood loss over several hours until other mechanisms can take over. Recall from the last section we talked about smaller blood vessels called capillaries and venules and arterioles. Here we see the vascular spasm is an adequate measure to prevent blood loss from these smaller vessels. The next step we get to is the platelet plug formation. In this diagram on the side, we might recall that megakaryocytes produce these tiny cell fragments that float freely in the bloodstream. Here we see those platelets are freely floating, but here they are adhering to the walls of that simple epithelium. So in summary, this platelet plug formation begins with the sticking of the platelets to collagen fibers, and we can see that in steps 1 and 2. And later on, we get a longer process that's going to be leading to the attachment of these platelets to the walls of the blood vessel, and then they're going to accumulate together. In other words, platelet aggregation. This is going to create what's called a platelet plug, shown in the lower right-hand corner. The third step in this process of hemostasis is blood clotting, also known as coagulation. This is where fibrin threads reinforce that platelet plug made in the previous step. So in this blood coagulation, we're going to be working with fibrin, and fibrin is going to be derived from fibrinogen, and that's going to make up a clot of insoluble proteins. And so ultimately what's going to happen is we're going to get a meshwork of proteins that traps other formed elements in the bloodstream, things like erythrocytes, leukocytes, platelets, and plasma proteins to form that clot. Substances required for clotting include calcium and also enzymes synthesized by liver cells and substances released by platelets or damaged tissues. So the idea is that when this clotting is occurring, there are chemical messages sent out in that process. So we're going to go more in depth into that releasing of chemicals, this clotting, this sort of cascade of reactions in which each clotting factor activates the next one in a sort of fixed sequence. The end result is that we are going to be forming the fibrin threads that allow the bleeding to stop. So let's look more in depth into this idea. This slide serves as an overview of this clotting cascade. In this diagram, we see that an intermediate step is the production of an enzyme called prothrombinase, and there's two ways we can get prothrombinase formed. One way is the extrinsic pathway, and the other is the intrinsic pathway. The extrinsic pathway of coagulation is initiated by external tissue damage. This intrinsic pathway of coagulation is initiated by internal vessel wall damage and first enacted by those platelets we discussed. How can we master this complex process? Keep in mind that the word extrinsic kind of sounds like external, so hence the external damage on the external part of the blood vessel. And when we say intrinsic, think of sort of inside the internal vessel wall damage. Notice that again, whichever pathway we go, the final pathway is that we produce these fibrin threads which stop the bleeding. Let's take a closer look now at this extrinsic pathway. In the diagram on the right, notice that we're following this pathway. So this extrinsic pathway is, again, initiated by damage to tissue that is outside the vessel. And this is a fast reaction. This is going to take place approximately 15 to 20 seconds after the damage. Why is this more quick? It's because you can probably observe in the diagram that there are less number of steps involved. So what we have in the extrinsic pathway is tissue factor, that is factor 3, thromboplastin, is released from the damaged tissues and it combines with factor 7 and also calcium to form a sort of complex. After this complex is formed, it then converts inactive factor X to an active form of factor X, or factor 10, so to speak. This factor 10 is then activated by either the intrinsic or the extrinsic pathway to lead to this final common step, the common pathway. The idea is, again, that we're creating either pathway in the substance called prothrombinase. Now let's take a closer look at this intrinsic pathway. Again, the word intrinsic meaning inside, so to speak. This inside or intrinsic pathway, when activation occurs, that's when the endothelium lining the blood vessel becomes damaged. These platelets come in contact with the collagen fibers in that blood vessel wall. These platelets then release messengers, so to speak, in the form of phospholipids. This intrinsic pathway is a little bit more slow. It takes approximately 3 to 6 minutes. And we have a variety of steps involved and a variety of clotting factors. And again, our goal is to produce the substance prothrombinase. Let's look at some of the steps that we'll see to arrive at that chemical. First, the platelets are adhering to that damaged vessel, and that's going to release factor 12, the X and the 2 there. Then this factor 12 converts to the inactive factor 11, and then to an active, making that factor 11 now an active factor. Then factor 11 is going to convert the inactive factor 9 to an active form of factor 9. And then factor 9 binds with the calcium and a platelet factor 3 to form a complex that converts inactive factor 8 to an active factor of 8. Lastly, we're going to get factor 8 converting an inactive factor 10 into an active form of 10. And this activated form of factor 10 is then going to lead to that substance prothrombinase. After we get the prothrombinase created, we're going to follow what's called the final common pathway. Under the presence of prothrombinase and the presence of calcium, we're going to have a chemical conversion happen. And the chemical conversion is that we produce a substance called thrombin. And so in the presence of thrombin, this calcium converts soluble fibrinogen into a insoluble fibrin threads. And so the general steps we see that are occurring here are that the active factor 10 combines with factor 2 and 5. It's also going to combine with the calcium and platelet factor 3. And that's what forms the prothrombin activator. So then prothrombin activator activates prothrombin to turn into thrombin. Then thrombin converts soluble fibrinogen into that insoluble fibrin. And again, with the presence of calcium, the factor 13 is activated, and this stabilizes and works with the fibrin monomers to create a sort of framework for that clot, so that the clot can form and effectively stop the overflow of blood loss. This diagram serves to overview kind of a summary of all the steps that we just talked about. Again, the ultimate goal is to provide a blood clot, a coagulation, so that there's no more blood leaking out of that vessel. So after those complex steps, we have formed a blood clot. And keep in mind, this blood clot is a temporary measure to stop blood loss through a damaged vessel wall. Then, to return things to a homeostatic balance, this blood vessel must be repaired, and that clot must also be eliminated. So there's going to be a couple steps to eliminating that clot, and that's going to include clot retraction and fibrinolysis. The elimination of this clot, the first step being clot retraction, that's when this clot is going to be forming actinomyosin. This is going to be a contractile protein within the platelets, and that sort of squeezes the serum out of the developing clot, sort of takes some of the fluids out of that. The effect of that is it's going to make the clot a lot smaller, and we can get the walls, the endothelium, the walls of the blood vessel can pool closer together. After these walls pull together closer, we have the fibrin strands get degraded. So when we say fibrinolysis, we're talking about the breaking down of those fibrin strands that once provided that clot. After the clot is extracted, then the repair begins. Fibroblasts and endothelial cells will repair that damaged blood vessel. So it takes certain special enzymes and vitamins to make this happen. And one of those is vitamin K. So to get successful blood clotting, it does require adequate vitamin K. This is going to be a fat-soluble vitamin, and we can see on the right side vegetables such as kale or cabbage or broccoli or cauliflower are all high in this vitamin K. This vitamin K is important for synthesizing 4 of those clotting factors we discussed. And how might we also get vitamin K? Interestingly, bacteria in our large intestine are able to convert some of this very high in fiber sort of foods and extract vitamin K from it. So really, vitamin K is almost a sort of waste product created by bacteria living in the large intestine. Typically, blood clotting is a beneficial process in the human body, but sometimes there's a little bit too much clotting. So we have to be careful to prevent unnecessary clot formation, and we also need to prevent those clots from spreading. Obviously, if there's a clot that's spread elsewhere, it might plug up a blood vessel and prevent the flow of blood. And if we prevent the flow of blood, certain tissues are not getting the oxygen and nutrients that it needs. So we have different types of anticoagulants that are present in the blood, and these anticoagulants can be natural in the bloodstream or synthetically imported. So the idea is that clotting only is going to occur when the stimulus for clotting is strong, such as at the site of a wound. We have 2 general terms for types of intravascular clotting. Sometimes these are too large of clots that have problems for the human body. One of those is going to be a stationary clot. That's a clot forming in an unbroken blood vessel. So that, in other words, that clot should not be there. If blood flows too slowly, that can allow clotting factors to build up locally and cause this unwanted coagulation. Hopefully, if a person has good health and good exercise and diet, this stationary clot will dissolve spontaneously or dislodge and travel. Hopefully, if it dislodges and travels, it might end up in the spleen where it's broken down, but unfortunately, it can dislodge and travel and clog vessels later on down the system. And then an embolism is a term for a clot, sort of air bubble or fat from broken bone traveling in the bloodstream. So, for example, a pulmonary embolus is found in the lungs. Interestingly, aspirin has a chemical effect on the production of these unwanted clots. With blood clotting behind us, we now turn our attention to blood groups, and then later we talk about transfusions. So this cardiovascular system does want to minimize blood loss, of course, and we talked about how clotting is going to prevent that. If there is a large amount of blood loss, then the patient must receive a transfusion, or also if there's a low amount of oxygen as well. So sometimes in a transfusion, this person is going to receive donor blood from elsewhere. So that's why we're looking at blood groups and to see how donation and receiving blood can occur. Blood groups are essential for the companies like the American Red Cross, such as donating blood. Also, interestingly, we also prove or disprove paternity through blood groups. We could link suspects to crime and also study some anthropology, history among races. So there's more than one function for having blood groups. Let's look at these blood groups. We have 2 terms, antigens and antibodies. Antigens are going to be located on the surface of the red blood cell. So let's say this large red dot on the right represents a red blood cell. And on the outside of this red blood cell are going to be certain biochemical structures. Here, I just happen to make them triangular in shape. Each red blood cell, or each group of red blood cells, has its own special group of antigens on the outside. So antigens are located on the surface of the red blood cells. And then Another term we should familiarize ourselves with are antibodies. Antibodies are going to be on the outside of that red blood cell. More accurately, I should say, it's going to be floating in the plasma, the blood plasma. So these antibodies are not naturally found on the red blood cell itself. However, it can interact with that antigen. So in this example here, these green antibodies are floating in blood plasma, and here what I'm drawing in green now is showing an antibody that has connected with an antigen. And notice how they kind of fit lock and key, so to speak. So what I'm drawing here is actually a bad situation. This is going to lead to agglutination and clumping, so to speak. In an ideal situation, perhaps a normal situation would be where we had antibodies that had a slightly different biochemical shape. And with this slightly different biochemical shape, we are going to prevent agglutination from happening. Well, that should not have happened, but hopefully you get the idea that in a perfect situation, antigens and antibodies should not be meeting up. Okay, so coming back to antigens, individuals have different types of antigens and different combinations. There are actually something like 30 different genetically determined blood group systems. But generally speaking, most persons are using the ABO system. In other words, blood type A, blood type B, blood type O. And then the RH system as well. That's when they say positive or negative. So let's look more into these blood groups. Here is perhaps a better drawing showing the presence of those antigens on the surface of the red blood cells. And then we see the plasma has the antibodies. And again, if you look closely at these antibodies, notice how they might or might not match up. So let's take, for example, a person who is blood type A. A person with blood type A means that their red blood cells have type A antigens on the surface of that cell membrane. In an ideal situation, that person also has plasma, which actually has anti-B antibodies. So be careful. Antigen and antibody sound similar, but notice that, again, a person with type A blood is going to have antibodies B. Why is that? Because notice that these sort of triangular hashes will not lock in with the circular features of the antigens. So that's an ideal situation. And let's look again now at the type B sort of blood type. Again, here's this B type antigen on the surface of the red blood cell. And by the way, these are going to be glycoproteins and glycolipids that are these indicators. Here is this antibody A, which should be in the plasma. Notice how these should not be linking up. If they did, it would be causing agglutination, and that would be unfortunate. Notice how in a type AB blood, the person has both A and B antigens present, but in their plasma, there should be no antibodies available. Lastly, our type O blood type, on the surface of that red blood cell is neither A nor B antigens, and this person does have both types of antibodies floating in the plasma. This slide serves as a summary or a text summary of the previous slide, showing visually how those antigens and antibodies are present. So in summary, the presence of antigen A or antigen B determines your blood type. And then these antibodies, also called agglutinins, are going to be these Y-shaped proteins of the plasma, and they're going to recognize and immobilize antigens of erythrocytes that they perceive as foreign. So when they perceive it as foreign, they agglutinate. They unfortunately clump up. Combining incompatible blood types causes antibodies of one type to bind to the surface antigens of the second type, resulting in agglutination, or aka clumping, of the erythrocytes. The effect of this is going to cause blood vessel blockage and inadequate circulation. And again, if these vessels are blocked, if there's not enough circulation, then the tissue cannot receive the important nutrients. In the lab, we do have similar activity to this, but let's go ahead and talk about this, or not. In this diagram, we're showing that process of agglutination. So here on the first row, we have a type AB blood type. Remember that their serum is going to contain A, B. Let's look at this. Here we go. This is a better diagram. Their type AB blood has both A and B antigens, but the serum should not have any, the plasma should not have any antibodies in it. In this diagram, what we're doing is introducing the bad antibodies. So in other words, a type AB blood does have A and B antigens. In this diagram, they're introducing antibody A, and they're trying to show agglutination and clumping happening. And again, that's a bad thing. In this diagram, they've shown a scientist, let's say, putting a drop of blood in a well, and that blood is type AB. That scientist is then putting antibodies B in there. Well, they're going to agglutinate. They're going to clump up, and that's a bad choice. Now, why would we do something like this? This type of activity allows us to find what the blood type is. So we can identify the blood type through what it agglutinates with. And that's one of the main activities we do in our first day of lab. Let's practice that again. We're going to test this unknown type here. Let's say we don't know what the blood type is. Now they tell us that it's type A. But again, let's say we put this unknown blood type into well number 1, and we also put that same blood into well number 2. In well number 1, we're dropping antibodies A into there. And we see that because the antibodies are agglutinating with antigen A's, that means, yes, there is the presence of antigen A on the red blood cell surface. Then we see in this well to the right, we have placed again that unknown blood sample. In that well, they placed antibodies B. And antibodies B have a different biochemical structure on these glycolipids, and therefore they cannot connect, they cannot agglutinate, and we see a normal sort of looking blood there. So this would indicate then that the person is a type A blood. Why? Because when we put type A antibodies in there, it showed that they did have the presence of antigen A. And that process repeats with a blood type B, where the reverse happens. And then lastly, we see a type O. We can tell it's type O because no agglutination happens. Why? Because those red blood cells have no antigens present on the surface. So blood typing is a way that we can test for the blood type using antibodies. This is a general summary of group interactions, and we've already talked about some of this. In other words, type A blood has the presence of the antigen A, and in their plasma should be the presence of antibody B. And now we move into a new topic, and that is who can donate to who? Well, let's think about this. Is it possible that a person who is type A, can they donate to a person who's also type A? Yes, because it's the same type of situation. Notice that this person can also work with type O blood. Now, why can we work with type O blood? Let's look at that. Well, notice that there are no antigens on the presence of that red blood cell to clump with these antibodies. So hence we get a compatible donor situation. Let's look at an incompatible donor blood situation. So again, let's say this person is a type A blood type. What would happen if we put blood type B into their bloodstream? Well, these antigens on the blood type for B would start connecting with antibodies A in that person. And when they start connecting, we would get agglutination. Agglutination leads to clumping, and that would be an unfortunate situation. So in this train of thought, we continue through each blood type, seeing who is compatible and who is incompatible with that blood type. In addition to the classification as a blood being type O or type B or type AB, etc., we also have Rh blood groups. And it gets its name Rh because this antigen was discovered in blood of a rhesus monkey. So the presence or absence of this Rh surface antigen, also called an Rh factor, determines a person's Rh blood type, in other words, positive or negative. So the presence of the Rh factor positively can classifies a person as Rh positive, whereas the absence of the Rh factor classifies a person as Rh negative. And again, we do want to have the positive, I should say, the proper matching of blood types as discussed in that previous diagram. And so transfusions can be problematic with these Rh groups. So we're going to talk about this transfusion and pregnancy in a short few. This diagram is just sort of a food for thought of blood types in the United States. We mentioned earlier that these blood types are determined by antigens, and these antigens' presence or absence is determined through genetics. And a student would not have to memorize this diagram, but it's sort of interesting food for thought, showing how blood types do follow along certain cultural identities. Our next slide talks about blood compatibility. And again, a student would not need to memorize this, but a student should know who can donate to who and who can receive from who. Some of it gets a little bit complex. There's some interesting anomalies in the donation and recipients of it, but generally speaking, try to think about what are the antigens present and what are the antibodies present. That is a good clue of who can donate and who can receive. Let's talk now about this hemolytic disease of a newborn. This relates to, again, blood grouping and how there can be a normal situation and how there can be an unfortunate homeostatic imbalance in the second pregnancy. So here we're talking about hemolytic disease of a newborn. So again, this only is in reference to pregnancy or giving birth. And keep in mind that during pregnancy, the blood from the mother and the fetus will have constant mixing of blood. So during pregnancy, if the mother's blood type happens to be Rh negative and her fetus's blood happens to be Rh positive, there can be a possible medical complication. Let's look into that. What happens is the mother may produce antibodies that counteract the fetus's blood. So if the mother, prior to pregnancy, has never produced antibodies to Rh positive blood, then the fetus may not have any difficulty. But if the mother has already previously formed antibodies to that Rh positive blood, then the fetus will be in jeopardy. So in summary, if there is a conflict in Rh grouping, the first pregnancy sometimes goes quite well. The second pregnancy can be problematic as those antibodies will attack, quote unquote, the natural antigens in the fetus because it thinks it is a foreign substance. So as we said earlier, an individual might have a low amount of oxygen in their bloodstream, or they might have lost a serious amount of blood. This might require a blood transfusion. And this is where medical personnel will have to first do a blood typing, and then perhaps next do a cross-matching. So typing is determining the type of blood. And we talked about that a couple slides back where we showed putting antibodies into unknown blood samples. After that blood type is determined, we might do a cross-matching, mixing of the donor and recipient blood to check for compatibility. Keep in mind that whole blood that we've been discussing is almost never transfused today, and it's generally not available. When a person donates a unit of blood, they sort of divide it into three main components: erythrocytes, clotting factors, and platelets. So then a person who is receiving a transferred blood is going to receive what's most likely called a packed RBCs. They're going to receive a large concentration instead of just the whole blood has all that plasma. So again, the transfer of the packed, whether it be whole blood or packed, is going to be used to treat anemia or severe blood loss. And so we have to watch out for incompatible blood transfusions, again, such as those antigen and antibody complexes. Lastly, we leave off with just some general good terms to know to get you prepared for quizzes and upcoming exams. So that concludes our chapter on blood of the cardiovascular system. What's coming up next is the heart. So keep an eye out for videos on that, and we will talk to you then. Happy studying.